Hello everyone, my name is Hubert and welcome to Teach Me channel. Today we'll be looking at biomedical science. How does PCR work? This is an exciting day because this is the very first day, very first video about which I'm doing on biomedical science, which I've never done before, so wish me luck. Okay, anyways, without further ado, let's get started. So, how does PCR work? PCR is a polymerase chain reaction. It is a way of amplifying DNA. It is a way of getting one strand of DNA and then essentially copying it, multiplying it to get lots of strands of DNA. That's what polymerase chain reaction does, and it is exciting because it has so many applications. It may be just copying the DNA, but as I will explain you in the upcoming videos over the next coming weeks, it's actually really important in any sort of molecular biology. It has so many applications. PCR has so many variations and so many applications, and that's important to know. Now, I will just cover the basic reaction, and I will explain it to you how the basic reaction works. I will not go too much into amplifications, and I will not go too much into the different variations, and I will not go too much into the different types of PCR, but I will be covering that in the upcoming videos. So, without further ado, let's get into this reaction. First of all, here we have a strand of DNA. Two strands, double-stranded DNA, and this is the target. This is what we want to amplify. This is what we are interested in. So, what do we need to do first? First, we need to split. We are splitting that double-stranded DNA into two single strands. And that step is called denaturation. And how do we split it? We split the molecule by increasing the temperature to around 94 degrees Celsius. That is enough to overcome the hydrogen bonds that are holding these two strands together, and therefore it split them into two. So after denaturation, we have two separate strands of DNA. And now we will copy them using the bases that are already there. The bases that are already there on each strand. How do we do this? Well, First of all, we need primers and nucleotides. Nucleotides are essentially these little bases that are freely floating around in the fluid, and they will be able to attach onto the existing template strand. And we need primers. Primers are important because they allow you to start. Primers give you the starting point, and primers allow you to pick what particular part of that long DNA sample you want. They allow you to pick the target, what I squared here in a box. They allow you to pick the specific parts of the DNA that you want without copying all the other stuff, the entire, genome. Okay, so after denaturation, we need to stick primers on. We need to do this. We need to stick the primers on, and to give us the beginning point. We have two primers. One is the forward primer, one is the reverse primer, and they both work in the opposite ways. I will get onto why that's important. But you need two primers that will attach themselves onto molecule in a process called annealing. I'll write it down. And we begin that process of annealing by lowering the temperature down to around 60 degrees Celsius. There are, by the way, some variation between those temperatures. Some people prefer using slightly different temperatures. But on a whole, annealing should be happening at around 60 degrees Celsius. Now, annealing is also, the temperature is also dependent in this case on the primers. The way you design the primers, by the way, I will go into primer design in some other video, but the way you design the primers will often dictate the ideal temperature for annealing those primers. This is the temperature where primers are actually in a best position, they have best conditions to form these hydrogen bonds to attach to their complementary part on the parent, DNA strand. Okay, so now we stuck the primers on. We have decided where our molecule is going to begin, where it's going to end. And then we are adding an enzyme called Taq polymerase, and we have our free-floating nucleotides. Now, Taq polymerase slides along those primers, and then it starts sticking on the bases, the nucleotide bases, which are complementary to the parent DNA strand. They start sticking on those nucleotides, and they sort of work their way along the new strand, forming a new strand essentially. Now we have two new strands of DNA. Two new strands of DNA, and so that makes us, that doubles what we had before. First we had one double strand, and now we have two double strands of DNA. So we essentially managed to copy that target using polymerase chain reaction. Now, you do this, you keep doing that, and you repeat those, you keep repeating that. You keep repeating that for about 35 to 40 cycles. Now, this is and then you double each time you double. So here you made 2, then you'll make 4, then you make 8, then you'll make 16, and so on and so on. You keep doubling with each cycle. So that's how we can go from literally one double-stranded piece of DNA to lots and lots and lots of copies. We can copy this so many times. Okay, so that explains the basic reaction, but there is one thing I haven't explained yet, and I will explain that to you now. And that's often something that people don't explain in these type of things. Now, so as I drew here, we have primers, and the Taq polymerase keeps working along the DNA and keeps making a new sort of complementary strand. Now, it keeps doing that. There is nothing to stop it as it stands. There is nothing to stop it. It's just gonna keep on doing that forever, right? So surely that's not gonna give us a target sequence. That's gonna give us something like a target sequence, but with sort of more DNA included, which is often called actually flanking DNA. Now, what do we do about this? How does that even work if we don't stop that Taq polymerase? I will show you on the next page. So here we are. 1 second. Okay, so here we are. And we have the two, in purple we have the two original DNA strands. In green we have the new ones that we just made, where they begin where the primer is. They begin where the primer is. But then they don't end. They keep going on. They keep going on, and sort of these bits are here called flanking DNA. That's the bits that we don't want. So how do we get rid of them? Well, we just keep doing the cycles. Like I said, we do this for about 35 and 40 cycles. So if we do if we keep repeating the cycles, what can happen is that the reverse primer will attach to here. Reverse primer will attach to the end of our target sequence, and then the enzyme, the Taq polymerase, will keep working this way. It will keep working this way, and that will give us like basically target strand. This is our first strand that will actually be exactly the target sequence. So then we can move on, and now we have a whole strand like so. And it is actually the exact copy of the target DNA. It contains only the DNA we want. And then the next one that's going to be made on top of this is inevitably going to look like this. It's going to be identical. And just like that, we made our target sequence. Target only. So that's how it can happen. Now, what's going to happen is that this isn't the only thing that's going to be happening. These primers will be attaching back to their original ones, make the very, very original DNA strands, making more of these green guys, and that's still going to be happening. So, initially, our DNA is not going to be so pure. The first sort of 10 cycles, it may not be ideal. It will contain some of these flanking DNAs and some of the targets, by chance. But over these cycles, by the time we get to the cycle 40, we will have vast majority of this DNA that contains only our targets and tiny minority of these guys which contain the flanking DNA. So this is how we essentially eliminate the flanking DNA. This is how it works. Okay, so this is it for today. This is how PCR works at the very basic, at the very core of PCR. This is what's happening. And I will make some more videos explaining the different applications and the different variations of PCR. But this is it for today. Thank you very much for watching the video. I really appreciate when you guys watch these videos. If you have any questions, I appreciate that this can be a confusing topic. So if you have any questions, just shoot me a question in comments below. I'm more than happy to answer to your questions. And if you like this video, give it a thumbs up. If you want to stay up to date on daily science, maths, and some biomedical science videos, hit the subscribe button below. Thank you again, and see you next time.